Today our topic is Bordetella pertussis. Bordetella pertussis, they are bordered Ginger bacillus, formerly known as Haemophilus pertussis. The morphology is the Bordetella species are small gram-negative cocobacilli with a slight pleomorphism measuring 0.2 to 0.3 mm by 0.5 to 1 mm. They appear singly or in pairs and in some clusters. On primary isolation, the cells are uniform in size, but in subcultures they become quite pleomorphic and the filamentous one, and thick bacillary forms are common. It is non-motile and non-sporing. Bipolar metachromatic staining may be demonstrated with the toluidine blue. Capsules may be demonstrable in the young, freshly isolated, and cultures only by the special stains. In culture, the films, the bacilli tend to be arranged in loose clumps with clever and clear spaces in between, giving a thumbprint appearance. The freshly isolated strains of the B. pertussis have the fimbriae. Cultural characteristics of the Bordetella pertussis. It is an obligate aerobe. The optimum temperature for the growth is 35 to 36 degrees Celsius. It does not require the X and B factors for its growth. Complex media are necessary for the primary isolation. The medium is common use. This is the bordered ginger medium, potato blade, glycerol agar. Primary isolation of the B. pertussis requires the addition of the charcoal ion exchange resins or 15 to 20% blade to neutralize the growth inhibiting effects. Potatoes, they impart a high starch content to the medium that neutralizes the toxic materials. Glycerol, they act as a stabilizing agent. Charcoal blade agar is useful medium. The plates are incubated at 35 to 36 degrees Celsius in moist environment. A sealed plastic bag. After incubated for 48 to 72 hours, colonies on the border ginger medium, they are small, dome-shaped, smooth, opaque, biscuit greyish-white, refracted and glistening, resembling the bisected peels or the macrodrops. The colonies are surrounded by the hazy zone of the hemolysis, confluent growth presence in the aluminium paint appearance. The subcultures of the B. pertussis may be obtained on the less exacting media. Example, the nutrient agar to which the charcoal or the starch has been added. Biochemical reactions of the Bordetella pertussis. It is biochemically inactive. It does not ferment the sugars, form the indole, reduce the nitrates and utilize the citrate or the split urea. It produces the oxidase and usually catalyze also. Antigenic constituents and the virulence factors of the Bordetella pertussis. It is a delicate organism. It can be killed by heating at 55 degrees Celsius for 30 minutes, drying and the disinfectants. Outside the body, it can survive for 5 days on glass, 3 days on cloth and few hours on paper. The organism is usually sensitive to the ampicillin and erythromycin, and these drugs have the reasonable therapeutic record. B. pertussis produces a number of factors that are involved in the pathogenesis of the disease. Agglutinogens. Bordetella possess the genus-specific and the species-specific surface. 14 agglutinogens associated with the capsular K antigen or the fimbriae. Factor 1 to 6 are found only in the strains of B. pertussis, all of which carry the factor 1 and one or more of the other factors. All three mammalian species of the Bordetella has common factor 7. The factor 12 is specific for the B. bronchosepticemia and factor 14 for the B. parapertussis. Agglutinogens promote the virulence by helping the bacteria to attach to the respiratory epithelial cells. They are useful in the serotyping strains and in the epidemiological studies. On the basis of the agglutinogens Bordetella carry, they are classified into various types. Pertussis toxin. They are also known as the lymphocytosis, promoting the factor, petusigen, histamine sensitizing factor, and the islets activating factor. They have a wide spectrum of the biologic activity. Pertussis toxin promotes the lymphocytosis, sensitization to histamine, and enhanced insulin secretion. It is expressed on the surface of the bacillus and secreted into the surrounding medium. This is the molecular weight of the 1,17,000 and has a classic A-B toxin dissociated into A and B subunits made up of 6 polypeptide chains consisting of the A subunit, toxic subunit, and the 5 binding subunit like S2 to S5, 2 S4 subunit that are present in the each toxin molecule. The B unit consists of the remaining 5 polypeptide chains, binds the toxin to the target cell and helps A unit to cross the membrane. It can be toxidized. Pertussis toxide is the major component of the acellular pertussis vaccine. It is apparently responsible for many of the clinical signs and symptoms of the pertussis as well as the relative and the absolute lymphocytosis observed during the clinical illness. Antibody against the PT is also protective in the animal models of the pertussis. It can be toxidized. The major component of the acellular pertussis vaccine is PT toxide. Antibody to PT can protect the mice against the intranasal, intraperitoneal, or intracerebral challenge. Outside the P. pertussis cells, the filamentous hemagglutinin and the pertussis toxin are found, which are secreted proteins. Filamentous hemagglutinin uh, the filamentous hemagglutinin and the pertussis toxin, they are secreted proteins and are found outside the P. pertussis cells. It is a protein that acquired its name through its ability to agglutinate the erythrocytes. It mediates the adhesion to the ciliated epithelial cells. The FHA is used in the acellular pertussis vaccine along with the PT toxide. Adenylate cyclase. All mammalian bordetellate but not B. avium produce the adenylate cyclase. At least two types of AC are known. Only one which has the ability to enter the target cell and act as a toxin. This is known as AC toxin. Heat labile toxin produced by all species of the Bordetella appears to be the cytotoxic protein. It is a heat labile toxin. It is dermonectotic and at the high doses. This toxin causes the fatal reactions in mice. Their role of disease is unknown. Tracheotoxin. This is a low molecular weight cell wall peptidoglycan monomer that has a specific affinity for the ciliated epithelial cell. The disruption of the ciliary function may also contribute to the secondary bacterial infection. The toxin also stimulates the release of the cytokine interleukin 1, which leads to fever. Lipopolysaccharide. It is present in all bordetellate and exhibits the features of the gram-negative bacterial endotoxin. Their role in the disease process is unknown. Pathogenesis of the Bordetella pertussis. The whooping cough is predominantly a pediatric disease. The whooping cough in 95% of the cases is caused by the P. pertussis. The parapertussis causes about 5% of the cases, and bronchosepticemia very infrequently, which is nearly 0.1%. Stages of the whooping cough. In human beings, after an incubation period of about 1 to 2 weeks, the disease takes a protracted course comprising the 3 stages: the catarrhal, paroxysmal, and convalescent, each lasting approximately 2 weeks. Prodromal or catarrhal stage. The first stage resembles a common cold with serous rhinorrhea, sneezing, malaise, anorexia, and the low-grade fever. The clinical diagnosis like catarrhal stage is difficult. During this stage, large number of the organism are spread in droplets and the patient is highly infectious but not very ill. This is unfortunate as this is the stage at which the disease can be arrested by the antibiotic treatment. Paroxysmal stage. After 1 to 2 weeks, the paroxysmal stage begins. As the catarrhal stage advances to the paroxysmal stage, the cough increases its intensity and comes on the distinctive bouts. Convalescent stage. After 2 to 4 weeks, the paroxysmal stage is followed by the convalescent stage, during which the frequency and the severity of a coughing gradually decrease, but the secondary complications can occur. Usually last 6 to 8 weeks, though in some it may be very protracted. Complications like subconjunctival hemorrhage, subcutaneous emphysema, inguinal hernia, or the rectal prolapse due to the pressure effects during the violent bouts of coughing. Respiratory, bronchopneumonia, lung collapse. Respiratory complications are self-limited. Neurological, like convulsions, coma, neurological complication may result in the permanent sequelae such as epilepsy, paralysis, retardation, blindness, and deafness. Whooping cough is one of the most infectious of the bacterial disease, and non-human contacts seldom escape the disease. The secondary attack rates are highest in close household contacts. The disease is often atypical. In adolescents and adults, they may present as bronchitis. They may serve as a source of infection in infants and children. Laboratory diagnosis. Blood changes in the disease are distinctive and helpful in the diagnosis. A marked leukocytosis occurs with relative lymphocytosis. Total leukocytic count is 20,000 to 30,000 with 60 to 80% lymphocytes. The erythrocyte sedimentation rate is not increased except when the secondary infection is present. 3 lab diagnoses are available. Isolation of B. pertussis by culture from a pernasal swab. Identification of the organism in a smear from the pernasal swab by immunofluorescence microscopy. Serological demonstration of the specific antibodies in the patient's serum. One is microscopy. The microscopic diagnosis depends on the demonstration of the bacilli and the respiratory secretions by the fluorescent antibody technique. Specimen collection and the transport. Though the disease is mainly in the lower respiratory tract, the organism can be recovered readily from the nasopharynx. The cough plate method. Here a culture plate is held about 10 to 15 cm in front of the patient mouth during about the spontaneous or induced coughing, so that the droplets of the respiratory exudate impinge directly in the medium. This has the advantage of the specimen directly inoculated at the bedside. Postnasal swab. Secretion from the posterior pharyngeal wall are collected with the cotton swab on the bent wire passed through the mouth. Salivary contamination should be avoided. A best postnasal swab may be conveniently employed. Pernasal swab. A sterile swab on the flexible wire is passed gently along the floor of the nose until it meets the resistance. Culture. The swab is inoculated immediately on the charcoal horse blade agar and bordered ginger medium, both with and without the methicillin or cepaxillin, and incubated for at least 7 days before being discarded as negative. The specimen may be transported in the Regan Lowy semi-solid medium if delay in transport or if for unavoidable situation. It nearly takes 35 to 36 degrees Celsius and 48 to 72 hours. Identification is confirmed by microscopy and slide agglutinating with specific antisera. Immunofluorescence is useful in identifying the bacillus in the direct smears of clinical specimen and culture. Detection of the bacterial antigen. Bordetella antigen may be detected in the serum and urine in test with the specific antiserum. Alternatively, the bacteria in the nasopharyngeal secretions are labeled with the fluorescence conjugated antiserum and examined by the ultraviolet microscopy. This method has the theoretical advantage and compared with the culture of detecting the dead Bordetella. Polymerase chain reactions. This is used for the detection of the Bordetella DNA in the nasopharyngeal specimen by the use of various primers with a sensitivity of the 80% to 100%. Rise in the antibody that may be demonstrated in the paired serum sample by ELISA agglutination. Complement fixation, immunoblotting, indirect hemoagglutination, and the toxin neutralization. Demonstration of the specific secretory IgA antibody in the nasopharyngeal secretion by the ELISA has been proposed as the diagnostic method in the culture negative cases. Treatment. B. Pertussis is susceptible to the several antibiotics except penicillin. Treatment and the quarantine. Antibiotics and immunoglobulins currently available are not effective for the treatment of the patients for the production of contact. Vaccination for the whooping cough. Phase 1 strain for the vaccine production. It is utmost importance. The vaccine is general use of the suspension of the whole bacterial cell killed by the heat or chemical. B. Pertussis act as an adjuvant for the toxoid like diphtheria and the tetanus toxoid produced by the antibody response. Adverse reaction. The Pertussis vaccination may induce a reaction ranging from local soreness and fever to shock and neurological complications like convulsion and the encephalopathy. The provocation of poliomyelitis is a rare complication. If severe complications such as encephalopathy, seizure, shock, or hyperexia develop following the vaccine, subsequent dose of the vaccine are contraindicated. Routine pertussis vaccine is not available after the age of 7 years as adverse reactions are likely and the risk of severe disease is low. Acellular pertussis vaccine containing the protective components of the pertussis, bacillus, VT, FHA, agglutinogen 123, first developed in Japan, cause for fewer reaction, particularly in the older children. Both whole cell and the acellular vaccines have the protection rate of about 90%. Thank you.